Okay, so I want to move on to the next session. That is the panel discussion. So the panel discussion will be chaired by the young Ellenberg, the EMBL. So most of you are familiar with this EoE meeting, so and GBI meeting. So you don't need any introduction of Jan, but maybe so several people are not familiar with this meeting, so I need a brief introduction of Jan. So Jan Ellenberg, I would like to do that. So I'm very honored to do that. So Jan Ellenberg is a senior scientist and the head of the cell biology and the biophysics unit at the EMBL, European Molecular Biology Laboratory in Heidelberg. And his research interest in cell division and nuclear organization, including the systematic analysis of mitosis, nuclear pore complex structure and assembly, as well as chromatin organization and the formation and the segregation of the nuclear meiotic chromosome. So he achieved very much scientific achievement. So, and also in addition to that, he has coordinated European and the EMBL effort to make imaging technology more accessible to researchers. And he is leading the foundation of the Global BioImaging right now. So, and also the EMBL Imaging Center. So he was also the scientific coordinator of the Global BioImaging. So without him, so there is no Euro BioImaging and no Global BioImaging. So I want to ask Jan to lead the panel discussion. So please welcome Jan. So.
Thank you very much, Shuichi, for this very kind introduction. Before I kick off the panel, let me just take a moment to really say how much I've enjoyed these 2 days of the Global BioImaging Exchange of Experience number 5 already. And especially during the pandemic. I think although we can sadly not enjoy the unsurpassed hospitality that we normally have from Naoto and colleagues in Japan, I think COVID-19 has made very clear how absolutely vital life sciences are and how vital it is that we support them internationally with international collaboration. I think this workshop is therefore very timely, and it's also really important to keep up this interaction and continue it very fruitfully. So I would, to start the panel discussion, I think it would be great if all the speakers could switch on their videos. So I think that was the technical format that we decided on. So these are the speakers from today that I think will be still fresh in everybody's mind. And I think for the keynote yesterday, I believe that Anne herself now will actually take over for the panel. Okay, so I can see Anna, Lisbeth, Bart, Wojtek, Anne is here as well. I just don't see Stephanie. Ah, no, there she is. Sorry. That's perfect. So we are ready to go for this. So I thought we are already a little bit behind time, and we've heard really exciting presentations yesterday from the member of Anne's group, but also today actually on really cutting edge image analysis in the area of high throughput microscopy, both in academia and in industry, and also of cutting edge deployments of artificial intelligence and machine learning for image analysis. And then on the other hand, we heard from Stephanie and Wojtek on the real brutal technical challenges to bring these advanced tools to the biologists who are producing the data and who have the research questions. And so I thought, following the discussion yesterday and today, there are maybe 3 broader areas that would merit some discussion and some input by the panel. And I think the first would maybe to talk a bit about infrastructure and services in bioimage analysis before publication, because this is the overall topic of this workshop. And I have some specific questions there for the panel. Then the second area I thought that would be worth briefly touching upon is more research and technology development. What are the new things and how can we make sure they quickly become useful and standardized for people? And then the third and maybe concluding topic I thought would be, what would this panel think should global bioimaging as a community and as a network focus on to really support the international community in bioimage analysis before publication? So these would be the three areas I thought that would be worth touching on: infrastructure, research, and what should global bioimaging do in the future. So that's a lot for 45 minutes, but so I would ask all panelists to make succinct comments if they can, and really shout out the most important ideas and most important points that they have. And so I would want to start with infrastructure and services. And I think we've heard a lot about different ways of analyzing data, but we also heard a lot about how hard it is to bring cutting edge tools to the garden variety biology user who actually wants to use them. So my first question here would be, how can we provide trusted open source bioimage analysis tools? And how can we do that via the cloud? I think the discussion we had, especially with our colleagues from Latin America, showed very clearly that that will be the future for the international community, to do this via the cloud. And so maybe we can start with that question. How can we come to agreed trusted tools and how can we remotely deploy them? And I would maybe ask the panel to briefly comment on this. And so maybe we go from the experts maybe to, well, I don't know in which order this makes more sense. So I just start in the order that I have the panel on my screen. And so I would ask Stephanie maybe to comment first, and then I will hand over to the next person.
Okay, so it's a hard question for me. Because
and you can also pass a question if you think that's a question you don't want to comment on, just pass it on.
No, I mean, it's maybe from the technical side, I would say we have to also deal a little bit with some legal aspects. Maybe bring everything to the cloud can be a problem. Maybe this is just one aspect I want to mention. Yeah. So, and this is, so I don't know how the development will go on with this national initiatives. So will they agree on putting everything to clouds, or will there be national clouds or whatsoever available? So I don't know. So it will be interesting to follow. That's the only comments, the only ideas, the thoughts that I have on that.
Mm-hmm. Mm-hmm. Yeah, no, it's a very valid point. I think here a magic solution is federation. I mean, that it's not going to be one monolithic cloud, but hopefully connected clouds where national systems have them. Yeah. So maybe the next person in my order would be Wojtek, to say something about trusted tools and how to deploy them remotely. And I think you really have in Australia a lot of experience with this, as you just showed.
Yeah, I think it's a bigger issue than what any one country can solve. I think it goes to the heart of the reproducibility crisis. And I'll help unpick that a little bit perhaps. So I think a lot of a big problem with web services for science is that a lot of the time they are very closed boxes, and there isn't a lot of transparency with regards to what's actually happening in the background. So what's the answer to that? Well, open source infrastructure, open governance, I think is also critically important with regards to how decision-making is made when a service is run, and how code is also accepted. I think that getting data out is just as important as getting data in. So allowing researchers the ability to export data is critical, because ultimately we're talking about researcher IP. And again, that comes back down to open source tools and governance as well. And then I think that there's the whole trusted data aspect to all of this. And, you know, that's critical to reusable data, to ensure that, well, sorry, to have data reused, you need to have a complete understanding of how that data was generated and what processing it's received to publication. And you know, there has been a significant amount of effort in Australia in this very space. For example, National Imaging Facility led a program on trusted data specifically for biomedical imaging, and that included aspects of making sure that there's an element of you know, quality assurance to any data that gets a tick of approval and is called trusted, as a simple example. But I think there's a lot to unpick with regards to just the word trusted and services.
Right. No, it's clearly a great challenge, but just, you know, I think we need to do this. And I think the effort you have led in Australia shows in principle a lot of things can be done in a remote manner. And so I think the perspective to do this even internationally in a remote manner is worth thinking about, to see what would really be needed to make it happen, what scale of resources and expertise would be needed, but also where do we need to standardize and connect people. Yeah, so I think this is a theme for the future. So maybe Anne could speak next from the panel.
Yeah, I feel my first comment should be a kind of big picture, very visionary thing, but I actually just have a laundry list in response to this question about deploying bioimaging in the cloud and also web-based apps. And so some of the pros of making a web-based app is that you're
unfortunately, and your audio, at least for me, is rather broken at the moment.
On multiple different platforms.
So maybe you can switch off your video to save some bandwidth for audio.
Okay, yeah, I'll try that. Sorry, the internet's not so good here.
Yeah.
Then secondly, I think another major advantage is anytime we're moving anything to the web or to the cloud, we're using technology that's developed for fields that have a lot more money than we do. Anytime that we can piggyback onto generic technology, I think it's really great. And cloud is definitely some place where there's a lot of developments, and there's no way that the resources we have can match what others are putting into it. The downside, I think one kind of minor one is in academia, we don't think about this so much. We are perfectly happy sharing all our data the moment we make it and aren't very secretive in my own lab and many other labs I know, but certainly in pharma and in other industries, it's not so feasible. So we have to think about security, which is something that for most academics is not a huge issue that we think about every day. Versioning, if we're using public resources that can change at a moment's notice, then an analysis you did yesterday on a web app might not be the same as one that's done the next week, and that can be really problematic for reproducibility. Most biologists wouldn't have, we would need to work on making it feasible for people to run their own instances if they want to use an old version of a thing, and then who pays for the computing. Either people either have to simply connect their cloud-based analysis to their own instances that they're paying for storage and so on, or somebody has to pay for it. And so that just this bandwidth and connection to servers, I think, is logistically kind of a frustration as well. So those are some of the challenges I see.
Thanks a lot, Anne. Very relevant. Since you mentioned industry, I will jump my my virtual order here and maybe hand over directly to our colleagues from Janssen. And so maybe for them, I would add an aspect to this question of how to deploy trusted tools in the cloud. With that question, can industry and academia actually share the same tools? Or does industry need to rely on closed proprietary tools?
Maybe I can take that one, Lisbeth. Yeah, so with respect to reproducible data science environments, Janssen has been we've really moved to AWS Web Services a lot. And in that environment, we relied historically or till recently a lot on tools like Chef, which do automated software configuration management. So they're basically it's producing recipes to set up a server, the installation of a server. That works well internally with respect to collaboration with academia. That's more challenging, and therefore I think, and that's also an internal effort, we see much more a move towards containerization, towards Docker being used. That certainly promotes exchange of data science environments, collaboration a lot. So I think that's certainly the future there. So for instance, also with Cell Profiler, I know there are Docker containers available on Docker Hub that we can use. So we're certainly using the latest version of Cell Profiler. Even full parallelized, distributed Cell Profiler is a tool that is available to actually parallelize the Cell Profiler processing of data. That is something that is more difficult to just copy paste in our environment because we are a bit restricted in AWS. We're not able to access plain vanilla AWS. We have only a limited set of services that we can use, and that's where we need to customize the setup a bit. But also that I think is a really great starting point. You can certainly leverage those setups and reuse them within our environments. I think containerization and then Kubernetes is also something, container orchestration platform that we're moving to more and more. So I think that is future there.
Thank you very much.
I think I can
Lisbeth, something to
add to that of course in the right framework that sharing the data between academia and pharma is of course desired. I can put the example of GEMCP, of the GEMCP consortium forward, and there we actually will and are doing that to try to combine data of different pharma companies together and make it public.
Yeah, I think that would be extremely valuable. I mean, in bioinformatics, there are similar initiatives also there in the pre-competitive space to share data that makes everybody able to mine a richer space and get more, far upstream pipeline insight into the whole drug development process. And I think if the image data would follow in the same direction, I think would be extremely valuable indeed. And also academically in the high throughput screening domain, data organization has been, because of the throughput and the volume, has been leading also in the IDR space. Yeah, so there is actually a lot of academic data there to look at. Yeah, absolutely. So I think Anna Kreshob would be the last panelist maybe to speak on this issue of powerful tool deployment remotely.
Okay, so here I kind of wanted to talk as someone who develops tools for interactive machine learning. And there what I want to mention is that, people take images to look at images. And this is something that we really have to keep in mind, because I've seen so many of these cloud initiatives where people are like, well, yeah, sure, we can put the tools together and invest a lot of time into making Java tools talk to Python tools as little Docker containers between themselves and ship things in between themselves. And then we submit the whole thing to the cloud and it runs there and computes. This is not what normal users want to do. What normal users want to do, they want to look at their pipeline results, right? No one wants to change a threshold value to 0.7, submit a batch job, come back after an hour and find that it should have been 0.65. Right? So people really want to interact with their images and to establish the pipeline that they want to run. After that, of course, you can run. Yeah, but you can run on the cluster or you can run on the cloud. The technology is not that different. That's why I was so happy to see what Wojtek was showing with this kind of virtual desktops. And whether it's a virtual desktop or a web-based tool is not so important, but we just have to keep in mind that all this cloud processing should really keep the interactivity of the tools that we develop. And this is the reason why people even still go to the desktop monolithic tools. The cloud is there, but still most of our deployments are on the desktop. And the deployment problem is in general still very difficult and somehow is not getting easier with years.
Yeah, I think that's very relevant. And I think that's also my feeling, that workflow configuration and customization is an iterative and interactive process, and then large-scale compute follows afterwards, and that can be implemented in different ways. But this first step is actually typically the most intensive with the end user, and you need to do this in a responsive mode, absolutely. Yeah, that's of course more challenging with cloud implementation. Okay, so maybe to finish the discussion on infrastructure, I actually just have a yes and no question for the panel, which is, do you think that an effective bioimage data analysis service, as we just discussed, can be analyzed globally for the global community or not? You know, is it too early to do this? So just a brief round of yes and no's from the panel. And so maybe we can go in the same order with Stephanie first.
I guess too early. No.
No. Then Anne, what do you think?
Too early, no. And maybe never, simply given the variety of use cases that exist in the world.
Okay. Our colleagues from Janssen, Lisbeth and Bart.
Yeah, quite early, I agree. Many challenges remain.
I agree there. I think difficult for us also to partner in, given some of the use cases, difficult to partner in those, I think.
Okay. How about Wojtek?
Oh, I absolutely think it's possible. I'm going to totally disagree with everybody.
I was hoping very much that you would say that.
Yeah, no, I think it's very hard. I mean, just sorting out. And we're not sorted out. It's just a project that we've got funded, and it's still lots of challenges. But that's not to say that we shouldn't try these things. And sorry, I've gone beyond my yes or no. but it's all in source. It's all in source code, and it is reproducible. And I think that's key.
Thank you, Anna.
Well, I don't really see why not, other than that people in general don't like to do things the way that others do them. But I think there is no reason not to. If we can solve the image formats problem, I believe we can solve anything. Thank
you. So we've got, I think if we lump the two Janssen participants into one vote, I think we have 2 yes and 3 no's at the moment. So I think clearly something to watch how we see the situation in 2 years' time. Yeah. So I wanted to spend a brief moment, and the organizers have to stop me if we are going too much over time for the schedule, but I think we started a bit late. I wanted to spend a few minutes on research and new technology with the panel, because I think we've heard a lot of exciting developments, and nevertheless also the difficulty of making it available. And of course, you know, without any surprise, the AI-based tools and machine learning-based tools are becoming very popular for the advanced developers and the advanced users. So my question regarding that would be 2. One is that going to take over everything? And so can we essentially forget classical feature extraction, classification, you know, formula-based designed image features and just do everything by AI? And if that is so, can this be made available to people in a safe way so that it avoids artifacts if people use AI that don't really know how these black boxes are working? So again, it's a complicated question, but I asked for brief answers. So basically, is it the future? That's a yes and no answer. And then the second is, can it be deployed safely to people who are not experts? So I think there are really 2 yes and no questions. And so maybe here, again, go in the order that we have established now, starting with Stephanie.
Okay, so it's again not so easy to say yes and no. I guess, of course, we are playing around with all these tools and we love it, and we are super, impressed by what's coming out there. And yeah, so, but on the other hand, it's not only images, but we also do a lot of spectroscopy, and there will always be fitting and normal application of models to our data and so on. So that will be a combination also in the future. And to give it to people, first of all, I would love that we really understand what's going on in this black boxes, at least that we can judge if it's reliable or not. And then, of course, we should give it to the people. Then they will anyway try it, because it's popping up everywhere. It's not only in our department, but also the informaticians are now doing it. And we have a special master program now available for the biologists for doing it. So it's there. So we have to deal with it and people will use it.
Thank you. So I guess that was a no and a yes to the two questions.
Yeah.
It's not the only thing, but it can be deployed safely. Yeah. Okay. Anne, how do you see this?
I would say that you have to define everything. Will deep learning take over everything?
Oh, of course, it's a provocative question a little bit. Yeah.
I really don't think like the entire world. But if we limit to segmentation, then I would say yes, deep learning will take over pretty much everything, and within a few years, I would hope. And that's a very limited part of image analysis is segmentation. So in cases where you need segmentation, I believe we'll be using deep learning. But I think biologists always need to measure things and count things, and not everything can be reduced to a segmentation kind of problem. I don't think deep learning should take over everything in the sense of, I treated these cells with a drug and these without a drug, you know, tell me the difference between the two, or identify the difference without any biological mechanistic understanding. I think those are where you get to your second question, which will it be available safely? So the advantage of using deep learning for segmentation is that we have the advantage of looking at the images and looking at the segmentation. So you try it on a new holdout set, and of course you can get the metrics of how well did it work, but you can also look at it by eye, and that's a huge advantage compared to people who study financial models and elections and all kinds of more murky machine learning problems. So I think that's what will make it very powerful and very safe to use for segmentation. But beyond that, it gets a little harder to wield this tool safely.
Thank you. Then how about Lisbeth and Bart?
Yeah, I can go first. Yeah, I agree with Anne concerning the segmentation. I think it will take over to a certain extent, and we are also evolving in that direction and setting up projects in that direction to immediately go from the images to the features. With the use of AI. And further than that, for us, we're also going towards more machine learning based predictions, where we can then actually set up these kind of virtual assays and use machine learning in that sense. And to your question, is that the future? I think so, yes. I think, well, I have to say yes there, because that's our project, and I definitely believe that as well. Yeah, to your second question, if this can be made available for everyone and if this is safe, it depends. Segmentation, probably, if well, losing the segmentation or dropping the segmentation to a certain extent, yes. These really large scale assay predictions is, yeah, a bit harder, I think, but to a certain extent, that will also further trickle further. So I think so, yeah, at a certain time, but that will be longer time frame, I believe.
Thank you very much. Maybe in the interest of time, we move on directly to Wojtek, unless Bart has an urgent thing to add.
I just wanted to say that with respect to machine learning models, that we're putting a lot of effort with putting the right governance structure around those, that if a model is being produced by data scientists, that they are properly validated, versioned, and only after this validation and evaluation, continuous evaluation of the models, they are released to the scientists, not just give everything to the scientists, but really in a controlled, safe way.
Yeah.
Because otherwise we will be throwing away the child with the bathwater, as we say.
Yeah. A very important point. Thank you for adding that. Yeah, absolutely. Wojtek.
Look, I think it is revolutionary, but I can't answer it from just from a microscopy or an imaging point of view. I could sort of I would have to answer this question by looking at, from my background, running a high performance computing facility and seeing the wealth of science that's benefiting. And I think that you do get revolution just through things happening quicker, right? And this is one way of doing that. Speaking to that and the second question, when I was still heavily doing the high performance computing aspects of my job, we would fret about the fact that bad science was being done faster. And this is the same. Bad science will be done, and that's okay. It's not, that's just normal. It's up to the scientific process to weed that out.
Right, yeah. And I think the point Bart made is very important, that the tools are well quality controlled that are provided, of course, yeah. Anna, this is a question for you, really.
Well, you know, in my little corner of the universe, it has already taken over. Yeah. Right? So there are these little blips of the classic techniques remaining sometimes, and sometimes you don't want to bother with training, and some problems are simple enough, but in the end, there's not much else left. Right? If you want to look at the cutting edge, there is nothing else left. And sometimes there is a non-deep learning tool being published at a computer vision conference, and everyone is like, look at that. So it's there's still a few, but it's really not many. And concerning giving it over to the users, well, there is still a deployment problem, which is still not solved, easier than 2 years ago, but we are still not quite there. It's still a pain, even inside the same institute. Once that is solved, because it feels like really more of a technical problem, you have to treat it like a normal part of your experimental pipeline, right? Design controls, look at what it's doing, validate in a way that is orthogonal to what it's predicting, validate on things which it has not seen, right? And then you can trust it. Well, I wouldn't say as much as you can trust biochemistry, but it's a method. It gives data out. You have to understand if the predictions are correct or not. If you can't see it for yourself, maybe you shouldn't be using it. And then if you can't validate it with an alternative method, if you don't use it just as a hypothesis generator, but as a final truth, well, that's not a good choice. But for everything else, I would say you will get there.
Thank you. That was really useful. So I have, again, a short question which would conclude the technology bit of our discussion. But it's important to me because I think both for using AI tools, but also for making data usable in the cloud, new data formats are needed. And the field of bioimage analysis really has been hampered for decades by difficulty of agreeing on common formats. And so my question to you as experts is, can we avoid this never-ending war of data formats to happen a second round, since we now have to change to do cloud and AI applications? Or can we use the opportunity to agree preemptively upfront and say it's already technically pretty clear what people should use, and that should be done by the community? Is there consensus, or is it going to be the same Cambrian explosion problem, and we will have another 20 years of not being as effective as we could be if we agreed on something? So very short answers, please, because we're running out of time, but maybe a quick round of opinions on this. Are we ready to agree here or not? Stephanie.
I guess we should be ready because we see that there's no escape from doing it. So, I mean.
Yeah, there's no alternative. Thank you. Anne.
Oh, I wish we could, but I'm also kind of skeptical.
But you would agree that there's no alternative. It needs to happen.
It would be wonderful if it happened, but on the other hand, we function okay in the kind of messy universe that we already function in. So I'm not going to say it has to happen. It would be lovely.
Okay.
But I'm.
Thank you. Lisbeth and Bart.
Yeah, I agree. It should happen. It really depends on the application as well. I can assume that the type of images that Anna needs, for instance, in 3D applications is completely different to everything else, everything what we use in 2D flat. So yeah, there will still be variations, but it would be a desire to go to the same format for the same type of application, definitely.
Yeah, I agree to that. I don't think we will ever reach the ultimate data format. There will always be new imaging modes and new data formats that go best fit to that new imaging mode. So I don't think there will be one final data format that we could use. And yeah, I have to apologize, but I really have to drop off. Actually, I have a meeting that I need to host.
Yeah, I'm aware we are very much over time, so I'm very sorry for that. Yeah, thank you for being with us, Bart.
Bye, everyone.
Yeah. Quick comment from Wojtek on this.
I think certainly in the large volumetric areas that we've heard a little bit about today, there's certainly an opportunity there.
Thank you. Anna?
Yeah, I agree. I think with the large volumetric images, there will be just really no choice. At least, there is no choice now, and I hope none will appear, and we can just stick with where we are now. And then the small 2D images will be like a zoo like they are now, but yeah, whatever, right? If people didn't feel the incentive yet, I don't feel like there's much we can do. Bioformats will save everyone.
Yeah. Good. Let's hope that at least in that area does happen, because I think it would really facilitate lots of downstream activities by everyone. The final question I wanted to address, an area to briefly discuss, and I think it will only take 5 minutes for the panel to give input on this. What should global bioimaging do in the future in this area of pre-publication image analysis? And I think from the discussion we already had over the last 2 days, the two sort of bigger topics I picked up was that on the one hand, several people commented that it would be tremendously valuable to have an international description of the image data lifecycle before publication, and to clearly point out what the technical issues are, what the technical solutions currently are, and which bits of this are the responsibility of local, national, and international funding agencies, so that one can come up with a reasonably coherent plan to say, if we want to make this happen, this is what it would take to actually do it. And I think that's sort of a classical GBI task, to coordinate, to collect information, and to distill from that what would internationally be useful. So I think this sort of international guideline would probably be very useful for people to go to their funders, also to instruct people for how to deploy best practice and for how to deal with things. And the other topic I picked up was training. It's a universal need, but specifically here, training in what is the best practice in this data lifecycle, and then in the most commonly used tools. And of course, data has been an area that GBI was active in training, and from its very beginning, there's already been several courses. And this topic certainly, I think, could be emphasized for training. So I don't think we need to discuss these two issues again. I think they are obvious that global bioimaging should do something there. But I would ask the panel, if you had a third wish, in addition to an international guideline and bespoke training courses on pre-publication image analysis, what would it be that you want global bioimaging to do as item number 3? So I'll start again in the customary order. Stephanie?
Okay, so for me, as I pointed out in my talk, maybe something like a provided platform for really also collecting the success stories, yeah, that we can tell people, okay, there's really a benefit, and this is an example that fits to your ideas, and this is why you should go for it. That would be great.
Thank you. And
I think a huge strength of this whole community is how non-dysfunctional it is. I think every once in a while I catch a glimpse of other scientific communities, and the sort of competition and just inefficiency of it is really discouraging. But I think in bioimaging we have a real strength that people love working together. People are willing to get along and try to pool resources and efforts together. So I think the best thing that an international group like this can do is provide venues for sharing what people are working on and what's working well. So for one thing, software, what software are people working on? I learned a couple of new tools today that I hadn't heard of before, so that's great, and I can go research those so that people can team up and join efforts on things, as well as sharing, so not just software, but best practices. And as Stephanie caught this very well, that if we can find some sort of venue for publishing how does my infrastructure work, getting down to the nitty-gritty logistics and practical details, I think that's what's really helpful for folks that are getting started, instead of the nice polished paper that shows the biological result and makes it seem so simple.
Thank you. Lisbeth?
That's also what I took along most of the talk, is what's happening behind the scenes and what we do not see in the nice polished papers. Which is the most informative exchange that one can have, that it knows that we know that it's not all shiny and works well, and that we can exchange that experience. I think that's really important. Yeah.
Thank you. Wojtek?
I'll just reiterate that I think standards and best practices are critical.
Thanks a lot. That seems pretty universal consensus. Anna, do you have a different wish?
No, I agree. I mean, the one thing that maybe I would as a tool developer appreciate, although I have absolutely no idea how to go around it, it would be nice to have a, you know, there where you have the standards and the best practices. It would also be nice to have like a list of the things for which we don't yet have best practices or standards. And this kind of an open problems where some might be just purely technical, some might be something that everyone does differently, and some might be just not solved with the current level of technology. So I think that would be interesting for many members and also for the potential new members.
Thanks a lot. Very valuable input indeed. I think with this topic, I would like to come to an end of the panel discussion. I would like to thank all the panelists and speakers, both for the great presentations yesterday and today, and also for the great discussion. I think that's very useful. I'm sure that our colleagues from Global BioImaging have taken good notes and will go through the recording also again to use this input for their future activities in this domain. So with this, I would hand back over to Naoto, our host, and give him the opportunity to wrap up and say closing remarks. And Naoto, I really hope that we can nevertheless have a meeting at some time in Japan and enjoy your famous and unsurpassed hospitality in Okazaki.
Okay. Thank you, Jan, and all the speakers. Before I close this meeting, I'd like to ask you all one thing. Please turn your video camera and give me your best smile. We are going to take a snapshot, screenshot of everybody. So Gleb, please tell me you have done screenshot.
One moment, I'll make it right now.
Mm-hmm.
Okay, it's ready.
Okay, done?
Yes, it's done.
Okay, thank you very much for your cooperation. Okay, before I close this meeting, I'd like to thank all the participants from the world who joined this meeting despite time differences, especially those who had to get up very early in the morning or stay up very late at night. I also thank excellent speakers for exciting talks, which stimulated us enormously. Yesterday, I was very impressed by that each regional bioimaging community is growing and being strengthened rapidly, including the one who is in the early stage. I feel that the global network is becoming denser and denser. This meeting was also indeed useful for global bioimaging community to think how and where we should move forward in the next few years, especially for promoting bioimage analysis and for sharing data, image data. I hope the current challenging situation of coronavirus will become better and we will be able to meet face to face to crystallize the idea we discussed and what we learned in this meeting. I don't know, I cannot predict when it will be, but let's hope that it will be soon. And I'd like to thank again all these people, especially Shuichi Onami and Jason Swedel for terrific scientific organization of this meeting, and Federica, Gleb, and Mariana, Joanna for smooth operation of this meeting. And on behalf of ABiS, Japanese bioimaging community, I thank GBI for giving us the opportunity to host this important and memorable meeting. Thank you very much.